Hello everyone, in this video we are discussing about the thyroid hormone synthesis, its secretion and its transport. T3 and T4 triiodothyronine and tetraiodothyronine, also referred to be as thyroxine, are considered to be as the major thyroid hormones. They are produced from thyroid gland. The thyroid gland is a true endocrine gland which is present caudal to the larynx, adjacent to the lateral surface of the trachea in all domestic animals, and it is present near the syrinx in case of birds. They are bilobed structures connected by a fibrous structure referred to as isthmus. The isthmus is very much prominent in case of ruminants and horses, whereas it is less distinct or less prominent in case of cats and dogs. And it is also noted that there is ectopic thyroid-like tissue which starts from the larynx up to the diaphragm in case of dogs and cats. So this, if you see inside the thyroid gland, it is made up of thyroid follicles which act as a basic functional and structural unit of the thyroid gland. This thyroid follicle has got a central compartment which consists of a gelatinous proteinaceous substance which is referred to be as colloid, which is surrounded by a group of thyroid follicular cells. So this spherical structure is referred to be as the thyroid follicle. And each thyroid follicle consists of a group of thyroid follicular cells, or these are also referred to be as thyroid epithelial cells, or they are also called thyrocytes. It should also be noted that the thyroid gland also produces another hormone which is a polypeptide referred to be as calcitonin, which plays an important role in calcium homeostasis, and it is produced from the cells called parafollicular cells, or they are also called as C cells, those which are present near to that of the follicular cells. So we do have one follicular cell here. If you consider this as another follicular cell, so near to the follicular cell there may be some parafollicular cell which produces calcitonin. So before going into the details of the steps that are involved in the thyroid hormone synthesis, first we will see the components that are involved in the production of thyroid hormones under three main broad categories. Number one, the materials or the raw materials that are required or that are involved in the thyroid hormone synthesis. Number two, the machinery. Number three, the control system which controls the whole process, or the components that are involved in the controlling system which will regulate the production of the thyroid hormones. So coming for number one, the raw materials. The most important raw material that is required for the production of the thyroid hormone is iodine. So the iodine which is present in the diet is absorbed from the gastrointestinal tract as iodide. And it is transported in the plasma which is being taken up by the thyroid gland. The thyroid gland has got an increased amount of this iodine inside it. The highest concentration of the iodine is present inside the thyroid gland. The remaining amount of the iodine which is not utilized by the thyroid gland is excreted in the urine. At about 20 to 25% of the iodine is taken up by the thyroid gland, at about 70 to 80% is excreted in the urine. So that is the reason why it is stated that the hormone which is mostly affected by the nutritional deficiency is the thyroid hormones. The second important raw material or the material that is involved in the production of the thyroid hormone is thyroglobulin. Thyroglobulin is a glycoprotein that is synthesized by the thyroid follicular cell. It is about 660 kilodaltons. It forms the backbone on which the T3 and T4 is formed. Number 3 is thyroperoxidase. The thyroperoxidase is also formed by the thyroid follicular cells, which plays an important role in the production of thyroid hormones in many steps. They are found on the apical surface of the thyroid follicular cell. They help in the process of iodination. It also helps in the process of oxidation. It also helps in the process of coupling and the formation of ultimately the T3 and T4. It catalyzes the different reactions ultimately forming the T3 and T4. Number 4 is the hydrogen peroxide. The hydrogen peroxide is also formed at the apical surface of the thyroid follicular cell by NADPH dependent through a flavoprotein referred to be as dual oxide. This is most important because these are the electron acceptor. Therefore, in the oxidation process, under the catalysis of the TPO, this plays an important role for the oxidation of iodide into an atom iodine or iodinium. We will see it later how exactly it occurs. The fifth important component is the tyrosine. The tyrosine is an amino acid which is present in the thyroglobulin chain, to which the iodide or the iodine is added upon. So as I already mentioned, iodine is the most important raw material because if you see T4, about 60 to 65% of the weight of the T4 is nothing but the iodine. About 50 to 60% of the weight of the T3 is the iodine. So this iodine attaches to the tyrosine, the amino acid. So now coming for the machinery. The area where the T3 and T4, the site of production is the thyroid follicular cells. So this is a thyroid follicular cell where we do have a lumen, an acellular part referred to be as a lumen, which consists of the colloid. The surface towards the lumen or the membrane can be referred to be as the apical membrane. So the surface facing the blood vessel. So if you consider this as the blood vessels is referred to be as the basal membrane, and these are the lateral membranes. So we do have the basolateral membrane and the apical membrane, and it consists of all the essential organelles. It consists of the nucleus, the DNA, endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, etc., all that are involved, that are essential for the production of different proteins like thyroglobulin and also the production of thyroperoxidases. And notably, it has got several transporters which are involved in the transportation of these substances that are important for the production of thyroid hormones. For example, on the basolateral surface, we do have a specialized transporter which is referred to be as sodium iodide symporter, NIS, sodium iodide symporter, which helps in the transportation of the iodide inside the follicular cell. So, the iodide, as I have already stated, is present in a higher concentration inside a follicular cell. Therefore, it is moving against the concentration gradient, and also it is moving against the electrical gradient. Therefore, it is an active transport and it is a secondary active transport, secondary active transporter, which utilizes the energy from the primary active transport that is again present on the basolateral surface, that is your sodium potassium ATPase pump. So, this is also present on the basolateral surface of the follicular cell. The second important transporters which is present on the apical surface is referred to as pendrin. It is an anion transporter which helps in the movement of the iodide which is taken intracellularly to the lumen. In the lumen, the production of T3 and T4 exactly occurs, and it has got all the organelles which are involved in the production of thyroglobulin, thyroperoxidases, and most importantly on the apical surface it also has DUOX, or referred to be as dual oxide, which is a flavoprotein which help in the production of H2O2, which is NADPH dependent, which is important for the oxidation of this iodide. Coming for the controlling system, the thyroid hormones production is controlled by number 1, TRH, thyrotropin releasing hormone, which is a tripeptide produced from the hypothalamus. The paraventricular nucleus and the median eminence produces the TRH, or the which is a tripeptide. This will act on the thyrotropes in anterior pituitary to produce a hormone called thyroid stimulating hormone, or TSH. TSH is a glycoprotein produced in the anterior pituitary by thyrotropes under the influence of thyrotropin releasing hormone. This is a glycoprotein, consists of alpha and beta chains at about 118 amino acids, and the other chain consists of the 92 amino acids in length. The most important is the beta chain, which is specific for the TSH. Most importantly, the TSH is mainly involved and it stimulates each and every step in the thyroid hormone production. Not only this, it also has the trophic or the nourishing effect on the thyroid gland. It increases the blood flow to the thyroid gland, and it also helps in hyperplasia and hypertrophy of the thyroid gland. Therefore, the TSH will act on the thyroid gland for the production of the hormone T3 and T4. So, number 1 is the TRH, number 2 is the TSH, and the third important controlling hormone is T3 and T4 itself. The circulating T3 and T4 can have a positive feedback mechanism or a negative feedback mechanism for the production of TSH, therefore its production can be controlled. So, now we will see in detail the different steps that are involved in the thyroid hormone synthesis. So, this is a thyroid follicle. We do have the basal membrane or the basal surface, the lateral, and this is the apical surface facing towards the lumen. And we already know the lumen consists of the colloid. And this is a blood vessel. So, now the first step that is involved in the thyroid hormone synthesis is referred to be as iodine trapping or iodide trapping. So, we have stated that the important raw material for the synthesis of the thyroid hormone is iodine. So, the iodine that is present in the diet will be absorbed from the gastrointestinal tract as iodide and it is transported in the plasma. So, iodide will come in the plasma. And it was also stated that the iodine concentration inside the follicular cell is more, therefore, this iodide will be moving against the concentration gradient inside the follicular cell. The process of taking in the iodide from the blood inside the thyroid follicular cell is referred to be as iodine trapping or iodide trapping. So, the iodide will move inside the follicular cell through a special transporter referred to be as the sodium iodide co-transporter or symporter. For one iodide, 2 sodium is also taken inside. So, when one iodide moves inside, 2 sodium also moves inside that of the cell. Now, the iodide is moving against the concentration and electrical gradient, therefore, it is works on the secondary active transport mechanism. Secondary active transport mechanism. So, this iodide which is taken inside a follicular cell will move to the lumen, and in the apical surface we do have another important channel or a transporter which help in the movement of this iodide to that of the lumen, which is called pendrin. So, it moves and it enters inside the lumen. So, this is the first step that is involved in the thyroid hormone synthesis. The second step that is involved in the thyroid hormone synthesis is thyroglobulin synthesis. So, thyroglobulin is a glycoprotein which forms the backbone for the production of the thyroid hormones. So, it has to be synthesized. So, it will be synthesized inside the thyroid follicular cell similar to that of the other proteins in a fashion in which the other proteins are synthesized. That is, there is the transcription of the specific area of the DNA which produces the mRNA, which will be translated. The glycosylation occurs in the endoplasmic reticulum. It goes to the Golgi complex where it will be packed, and after production they will be exocytosed out to the lumen. So there will be transcription. So translation outside, it goes to the endoplasmic reticulum, glycosylated, then it is a glycoprotein. It goes to the Golgi complex, it comes out as a vesicle inside which we do have the thyroglobulin, and it will be exocytosed. Through exocytosis near the apical membrane, it moves inside the lumen. So in the lumen we do have the thyroglobulin. So consider this as a thyroid globulin, which may be made up of more than 2000 amino acids. The most important feature of this thyroglobulin is that it consists of several types of amino acid, in which the amino acid which is of importance in the thyroid hormone synthesis is the tyrosin. So it will have tyrosyl residues. So these are the tyrosyl residues. There may be 100 to 200 tyrosyl residues that are present in a thyroglobulin molecule. The third important step, that is referred to be as oxidation of iodide. So the iodide that is present in the lumen has to be oxidized before it is attaching to this tyrosyl residues. So 2 iodide will be oxidized into iodine or iodinium or elementary iodine or the atom iodine. So for simpler we call it as iodine. So the iodide will be oxidized. So there will be oxidation to iodine, that is there will be loss of electrons. So this process will occur with the help of H2O2. We have already seen the H2O2, hydrogen peroxide, will be produced in the apical surface by a flavoprotein called dual oxide, which is NADPH dependent. So this hydrogen peroxide acts as an electron acceptor. This reaction is catalyzed by thyroid peroxidase or just peroxidase or specifically thyroid peroxidase. Therefore, oxidation of iodide into the iodine is mediated by thyroperoxidase, or thyroperoxidase is important for the oxidation of iodide into iodine, which occurs near the apical surface of the follicular cell. Only if this iodide is oxidized, it can link to the tyrosin residues in the thyroglobulin chain. Now the third and fourth step that is involved in the thyroid hormone synthesis is iodination or organification. That is, there will be binding of the iodine to the tyrosin residues. The process of binding of this iodine to the tyrosin is referred to be as iodination, or it is also called organification. So now, for example, if this is one tyrosin residue, if it is attached with, so it can attach with or link with iodine. So here we have one iodine attaching to the tyrosin residue. It is also possible that this another tyrosine residue can covalently link with 2 iodine, and it can again with 2, or it can be with 1, or again here in this example it may be with 2. Now, if a tyrosine residue is attaching with 1 iodine, then it is referred to be as monoiodotyrosine or MIT or monoiodotyrosine. Mono refers to 1, iodo refers to the iodine, tyrosine is the tyrosine residue on the thyroglobulin chain. Therefore, or in this example we can see one tyrosine molecule is attached with 2 iodine, so it is called diiodotyrosine or DIT. So, di refers to 2, iodo refers to the iodine attaching to or binding to the tyrosine. So, this through iodination or organification process, there is production of monoiodotyrosine and diiodotyrosine. Again, this process is also under the influence of, or it is catalyzed by, the thyroid peroxidase, or thyroid peroxidase also plays an important role in attaching the iodine to that of the tyrosine molecules. The fifth step is referred to be as coupling. Coupling refers to the linkage or the fusion of 2 organified or linked tyrosine residues. So, here, so we have seen there is formation of MIT and DIT. So, what does the coupling reaction mean? There will be fusion or the linkage between these 2. Again, it is under the influence of thyroperoxidase, or again thyroperoxidase helps in the linkage or the fusion between the 2 organified or tyrosine residues to fuse with each other. So, whenever it is fusing, for example, in this example, so here we do have one MIT which is fusing with DIT. So, whenever the MIT fuses with DIT, we get T3, that is triiodotyrosine. For example, if these 2 fuses, so here there is DIT and DIT. So, DIT fuses with DIT, then it produces T4, that is tetraiodotyrosine, also referred to be as thyroxine. So, this is how the T3 and T4 is produced, and to be noted, it is still present in the lumen. And what does colloid here mean? Colloid is nothing but which consists of the thyroglobulin, in which there is formation of T3 and T4. So, therefore, it is a proteinaceous substance present in the acellular compartment, that is the lumen. So, now coming back, how the T3 and T4 is produced. The number 1 step is referred to be as iodine trapping, wherein which the iodine that is present in the blood is taken inside the follicular cell through sodium iodide symporter or co-transporter, which is a secondary active transport mechanism in which 2 sodium and 1 iodide enters inside a cell. The iodide that is formed goes to the lumen through the transporter, that is pendrin. The second important step is the thyroglobulin synthesis. There will be the transcription factor that is produced, the portion of the DNA will be transcribed. There is production of mRNA, there will be translation, there will be production of the amino acid chain, which consists of mainly the amino acid tyrosine. There may be 100 to 200 tyrosine residues in a thyroglobulin chain of amino acid, which will be exocytosed inside the lumen. The third important step is the oxidation, where the iodide should be oxidized into iodinium or atom iodine or just iodine or elementary iodine. Only the elementary iodine will be attaching or linking to the tyrosine residues on the thyroglobulin chain, which will be with the help of hydrogen peroxide, which is catalyzed by thyroperoxidase. Fourth is the iodination or organification process, in which there is linkage of the iodine to that of the tyrosine residues, again with the help of thyroid peroxidase. Number 5 is the coupling. So, due to the organification process, there will be production of MIT and DIT, that is monoiodotyrosine and diiodotyrosine, which will be fused to form T3 and T4, which will be stored in the colloid inside the lumen. So, we can put number 6 as storage, that is, they are stored in the colloid. Sometimes in the examination, the question is also asked like this: the only hormone that is stored extracellularly is the thyroid hormones. Now, for the release, the 7th step is the release or the secretion of the thyroid hormone. So, now the thyroid hormone is present in the lumen. So, if it has to be released, it should first be taken from the lumen inside that of a cell. Therefore, there will be formation of an endocytic vesicle under appropriate stimulation, or whenever there is in need of thyroid hormones, that is T3 and T4, whenever there is stimulation from TSH, that is the thyroid stimulating hormone, whatever the thyroglobulin now present in the lumen, in which there is T3 and T4 that is formed, will be endocytosed inside. So, initially it was exocytosed for the formation, now for the release there will be endocytosed. So, it forms an endocytic vesicle, inside which we have the thyroglobulin chain, in which we have T3 and T4 which is fused. This endocytic vesicle will fuse with the lysosomes that are present inside the follicular cell. The fusion of the lysosome with the endocytic vesicle causes the release of lysosomal enzymes or hydrolytic enzymes, which will cut the appropriate sites. So, whenever it is cut, so we do get T3, so it is in blue, a thyroglobulin chain consisting of tyrosine residue attached with iodine 2. So, this is T3, or we can also have T4, and also sometimes we may also get MIT and DIT as such without fusion. So, they have not fused, but as such we may get MIT and DIT. And this T3 and T4 that is produced will come out to the blood circulation. And we do have a specialized transporter that is present on the basal surface, which is referred to be as monoconjugate carboxylate transporter. So, MCT, monocarboxylate transporter family, that is present on the basal surface through which the T3 and T4 moves out into the blood circulation. So, the MIT and DIT that is produced may be acted upon by the deiodinase and the iodide that is produced may be used, or it may be recycled and it may be reused for the production again. And it should be noted that the ratio of production of T3 and T4 varies. It is stated that the ratio may be anywhere from 1:20 as an average. However, in case of domestic animals, you have a very huge range of 1:15 to 1:45 in different domestic animals. However, as an average, we can consider as 1:20. T4 is produced in an increased quantity rather than the T3. However, it is less active. So, it is produced more, you can put it as produced more, active less, it is less active. However, again it has got a longer half-life. The half-life of T4 is about 2 to 6 days. Whereas T3, production is lesser, but it is more active than the T4, more active, and it has got a less half-life, that is than compared to that of the T4, it is about 16 to 48 hours. So, once produced or once it is released into the blood circulation, it is carried by attaching to number 1 TBG, thyroid binding globulin. So, thyroid binding globulin is produced from the liver where the thyroid hormone attaches to. So, it should be noted that the thyroid binding globulin attaches more, has got more affinity for T4, that is at about 70% of the T4 is travelled in the blood by attaching to thyroid binding globulin, and about 50% it attaches to the T3. The second binding protein for the thyroid hormones is albumin. So, it can also attaches to the albumin, but the notable difference is that albumin attaches more to the T3, at about 50% of the T3 is attached to the albumin and it is transported. The third important binding protein for the transportation in the blood for the thyroid hormone is transthyretin. So, trans refers to the transportation, Thi refers to the thyroid, retin is the vitamin A derivative retinol. It is also referred to be as prealbumin, or it is also called thyroid binding, prealbumin, where about 10 to 15% of the thyroid hormone attaches to it and it is transported in the blood. So, the amount of the T4 in case of domestic animals in the blood varies from 15 to 45 nanogram per ml, T3 will range from 0.5 to 1 nanogram per ml. There may be a very little percentage at about 0.03 percentage of the thyroid hormones that are unbound. Those are referred to be as free hormones, which are FT4 and FT3. Free T4 and free T3 are the unbound form of the thyroid hormones. T3, we have already seen, is the most potent hormone. It is about 5 to 10 times more potent than the T4. But T4 is produced in a very large quantity. Therefore, in the peripheral tissues, the T4 is converted into the T3 with the help of iodinase or 5' iodinase converts the T4 into the T3 in the peripheral tissues. Therefore, T4 is also referred to be as prohormone, because much of the T4 is converted into the T3, which is the most potent and the active form. Coming for the controlling systems, how T3 and T4 is regulated, we do have the TRH, that is a tripeptide produced from the hypothalamus, which will act on the anterior pituitary on the thyrotropes, and it helps in the production of thyroid stimulating hormone, which is the glycoprotein. Thyroid stimulating hormone acts on the thyroid gland, and it helps or it stimulates each and every step that is involved in the production of thyroid hormones. Number one, it increases the iodine trapping, it increases the thyroglobulin synthesis, or it is helpful in the thyroglobulin synthesis. So, it acts on the follicular cell. We do have a receptor on the thyroid follicular cell for the TSH, and it acts via GPCR. So, it is a G protein linked coupled receptor. So, we do have usage of GS stimulatory pathway, where there is production of cyclic AMP, the secondary messenger, and this cyclic AMP will phosphorylate many proteins, which will produce the transcription factors which act on the nucleus, which will transcribe and helps in the production of thyroglobulin. And it also increases the oxidation process, the organification, the coupling reaction, the endocytosis, and also the release of the T3 and T4 hormones into the bloodstream. It can also act via GQ pathway, that is, it utilizes phospholipase C and diacylglycerol and calcium as the secondary messenger. Therefore, this pathway is utilized for the production of the thyroperoxidase and also the hydrogen peroxidase, which is again we have seen it is most important in oxidation and organification processes. And T3 and T4, that is free T3 and T4, which is unbound, can also have its effect on the production of TSH from the anterior pituitary or the TRH from the hypothalamus. Whenever the concentration of T3 and T4 is decreased, it has a positive feedback mechanism so that there is more production of the TSH, or whenever the T3 and T4 is more, it can have a negative feedback mechanism on the TSH, therefore it decreases the production of TSH. Therefore, depending upon the concentration of T3 and T4, it can either have a positive feedback mechanism or a negative feedback mechanism on the anterior pituitary, so that it controls the production of